Yeah, I see a bunch of UMass people. I'm going to try and single out as many folks. Thank you guys for coming out and attending. Everyone, thank you, Fanbin, for the introduction. As Fanbin mentioned, our lab, I'm going to talk sort of broadly about what our lab does and some of the really cool projects that we have. You can kind of divide it up into sort of four main categories. The first of which I'm not really going to talk about too much today. We're doing some interesting work on developing alternative animal models to understand foodborne viruses, and in particular, we're interested in the role of gut bacteria in viral pathogenesis. So not really going to talk too much about that today. I think we'll talk about a number of projects sort of related to the next sort of three categories. The first one is upstream sample preparation of how you handle a food. So let's say you take a food or an environmental sample. How do you get from that to like a few microliters that you're going to load into a PCR reaction? That's still a very big area of research, especially for viruses. We have a number of really exciting projects about detection technologies for these viruses, as well as other pathogens in foods and the environment. And then the last sort of area is looking at controlling these viruses. So trying to understand and improve disinfectants to better inactivate these viruses, as well as the consequences of not adequately inactivating these viruses and the role that the viral structure can have in that. So you have a lot of really cool work. And we have, hey there. um, our lab focuses primarily on, hey there, kid. Um, so foodborne viruses are a leading cause of foodborne illnesses in the US as well as globally. And the bottom line from this is that they're extremely prevalent. I always like to point to an example of an estimate the CDC did that if you were to take sort of live the average life expectancy and ignore any host-related factors, each one of us would get norovirus 5 times. And specifically within foodborne viruses, we primarily work on norovirus because it's the leading cause of foodborne virus-related illness, as well as the single pathogen that is the most responsible for foodborne illnesses in the US and globally. And it is extremely prevalent. So even though it's the leading cause of foodborne illness, it's really about 30% or below of norovirus cases are foodborne. And then there's a whole other host of them that are person to person and environmental as well. So it's a very, very prevalent pathogen. And there's a number of properties that noroviruses have that sort of explain why they're so prevalent. So that make them also in some ways challenging to study, and our lab tries to address a number of these. So in particular with human noroviruses, it only takes a handful of viral particles to get somebody sick, so it has a low infectious dose. These viruses are incredibly stable, so they can survive on common touch surfaces and other surfaces for weeks to months. And then on top of that, they're also very difficult to inactivate. So a lot of the active ingredients of a lot of the commonly used disinfectants that you may sort of have aren't ideally effective against these viruses. You also have asymptomatic shedding of these viruses. And one of the other things that makes it a challenge in particular for vaccines, as well as it also affects disinfection that we'll talk about a little bit later, is that these are very, very diverse and they evolve very, very rapidly. They're sort of tuned into the evolutionary strategy of we're going to go in, we're going to create a lot of virus, and we're going to have very error-prone replication and create a lot of different variants of the virus, so that we can quickly escape host immunity, so that hosts don't develop immunity. Traditionally, it's also been a challenge in terms of being able to cultivate these viruses in vitro, which will come into play sort of the first thing I talk about as well. That, as you can imagine, if you can't grow it in the lab, it makes it very difficult to study. We do have a system now. It's a little bit imperfect and still has a number of limitations, but we do actually now have an organoid-based system for cultivating the virus, but it still is in the process of being improved, and it is limited in what you can do with it, despite being very, very revolutionary. So sort of the first thing that I was alluding to is, that you can't really grow these viruses feasibly for detection. So, one of the things our lab really focuses on is being able to detect these viruses in the environment. Now, one of the differences between detecting viruses in the food and the environment compared to like somebody who's shedding virus and just has a bunch of diarrhea and vomiting, like they're a factory for viruses when they're clinically presenting. Stool contains a high level of viruses, so it's pretty easy to detect. Whereas when you have contamination of the viruses in foods in the environment, it may not necessarily be at very high levels. And so you kind of have this needle in a haystack problem of you have this very large food sample and you have a handful of viruses that'll still get somebody sick if they eat the food. But again, it's sort of needles in a haystack. How do you get just those viruses out and get all the others, separate them from all the other food or environmental sample that you have? Traditionally with bacteria, you would do something called a selective enrichment step where you would take your food, stomach it, add media, grow it up overnight, and then presumably the pathogen you're looking for is going to grow to a higher level and it's going to be easier to detect. You can't necessarily do that with viruses for a number of reasons in terms of feasibility with time, cost, as well as just the host cells might die depending on what the food is. So it's not really realistic or feasible for detection of viruses. So essentially, you're forced to try and concentrate these viruses from the foods. And there's sort of two general ways that strategies that people will use to try and do this. The first one is a set of techniques that are based more non-specifically. I like to call them non-specific methods, where you're sort of exploiting the homogeneous nature of viruses, so their size or the fact that they kind of behave like a protein, so you can do polyethylene glycol precipitation. However, the problem with those non-specific methods is you tend to co-concentrate a lot of other crap from the foods that makes it that might inhibit your downstream detection technique. And alternative to that, there's more specific methods. Probably the most commonly known one or commonly used one is immunomagnetic separation, where you have antibodies specific to the virus that'll bind the virus, and those antibodies are attached to magnetic beads, and you can use a magnet to pull the virus basically out of whatever sample you have, wash away what's not, what doesn't bind to the antibody, and then elute the virus off. Problem with that is a lot of these methods tend to be costly. They tend not to be able to scale up to like larger sizes that you would necessarily need, especially in a food production environment. And they also can lack efficiency. So usually if you're above 30% capture or concentration, it's considered really, really good. ISO considers about 1% to be acceptable in terms of concentrating the virus, which is, again, not ideal. So sort of our first project is related to trying to concentrate these viruses from foods. It sort of originated from sort of the first thing that I talked about of some of the work that I was involved with as a PhD student, where we were looking at interactions of these viruses to bacteria. And one of the things we noticed, like we were looking more interested in a pathogenesis sense in terms of the aims of that project. But one of the things that we kind of noticed when we did that is that a number of these bacteria can bind the virus at a pretty high efficiency. So if you recall, I just mentioned 30% capture is considered really, really good. And you can see here you're upwards of 80% capture with some of the bacteria here for some of the viral strains. So we kind of had the thought, why not just replace, magnetic beads that have the antibody on it with bacteria as a capture reagent, which would presumably have a lot of benefits because bacteria are cheap, right? You can just grow them up very cheaply to whatever amount you need. They're potentially scalable, again, because you can grow as much as you want. And they also have an inherent size and surface area advantage. So why not replace beads with bacteria? And one of the other experiments we did in the process of this work sort of explains why it might be a little bit of a challenge. And that's that bacteria are living things. So depending on how their environment or culture conditions they can potentially dramatically display different binding activity, which for a detection or diagnostic technique is not ideal. So how did we address this? We decided to sort of take a trick from the biotechnology playbook, where we did something called cell surface display to try and guarantee that we'll at least hit a certain level of capture regardless of growth condition. And so we use something called the ice nucleation protein, which is a protein from Pseudomonas that essentially embeds itself in the bacterial membrane. And so you can clone a sort of affinity ligand on the sort of C terminus of it, and it'll present that affinity ligand on the surface of the bacteria. And so that's what we did. We basically took a number of peptides reported to bind noroviruses, clone them with a linker to sort of the C terminus end and express them in E. coli. So you're presenting these anti-norovirus peptides on the surface to capture the virus. This is based off of work where they're trying to use this technology to purify water and remove poliovirus. But in our case, we're trying to apply it for actually detecting virus. And we see some really exciting preliminary results. So here's G11, which is one type of strain of noroviruses where you see pretty strong capture rates with some of these. I'd like to shout out Catherine Wu, who I think is on this call. She did really cool work, and she's continuing to do really cool work in this space. So this is G11, the sort of lab type strain. And again, keeping in mind that 30% is considered very, very good. And again, you have a cheaper, more scalable type of capture reagent, which is really exciting that you see these really high efficiency capture rates. Similarly, we see really, really high rates with G24 Sydney as well, which is pretty exciting. So yeah, so we're really excited about the bacterial capture and really going to continue that because I think it really could become a valuable reagent in terms of this upstream sample preparation step, which still I think is the roadblock for most more routine detection of detection of viruses in the environment. Another sort of area of this sort of concentration field that we're working in is looking at magnetic liquids. There's two that I'm going to talk about today. There's magnetic ionic liquids, and which are hydrophobic molten salts that are extremely stable at room temperature. And they also, as a liquid, will display paramagnetic behavior. So you put a magnet, you pull them sort of out of solution, similar to like you would with magnetic beads. So it makes it more ideal for low resource or portable in-field settings. And the fact that it's flowable potentially allows it to be more integrated into automated portable systems. This is in collaboration with some folks at Iowa State who are, chemists and concentration scientists who synthesize these magnetic ionic liquids as well as the other magnetic liquids and send them to us so we can evaluate them for viruses. A lot of the work that I'm going to show you is done by recent graduate Sloan Stouffer, and I don't know if Lily is on the call, but Lily Syed as well also is doing some really cool work with this. So how do you actually use these magnetic ionic liquids? It's very similar in terms of a protocol to what you would do for immunomagnetic separation. You have your eluate or food sample that potentially contains viruses in it. You add your mils, you incubate the mils that'll presumably bind these viruses. You use the magnet to pull out and capture the mils. You remove the supernatant, wash, then you end up eluting the virus off those mils, extract the RNA, and quantify. And so with the mills, we see, again, pretty promising capture and recovery. One thing I would note as you look at the data is we have sort of two different metrics that we use. So one is called capture. That's essentially looking at how much virus is removed from the supernatant. So it's just purely how much virus that those mills are capturing. And then there's recovery, where you have not only that capture step, but how much can you elute off of the mills and get back. And so that's just like a heads up and a note that that's what the difference is between those two. And one of the things we see with these different mills that each have a different sort of metallic core that helps enable them to be magnetic is that you can see pretty strong capture for both the viral particles on the left as well as purified RNA on the right, which I'll talk about in a little bit. We not only saw this work really well in buffer, but one of the things, especially for charge-based capture, is that you really do want to evaluate it in more complex systems. And so here we use lettuce. We're in the process of starting projects to try and look at other potential rinsates or eluates. And again, we see pretty strong, we see some dampening effect of the lettuce rinsate, but not too much. You still see reasonable enough capture and recovery. It is interesting that for some of them, especially the elution buffers seem to really dampen the recovery rate, which is something that we're trying to optimize further. But it does seem like it still shows promise when you look at least in lettuce. We're in the process of starting to think about berries as well as eventually oysters, which are always a problem. One other question with these technologies is it going to capture infectious virus, and is it potentially degrading the virus as well? Again, because people haven't really done work with these magnetic liquids, you wouldn't necessarily expect it to have an antiviral effect, but you don't know. And luckily, as we found out when we sort of looked at capture rates or recovery rates with plaque assay compared to what we were seeing with PCR, they're very comparable, which suggests that no, the mills are not degrading the viral capsid, which is always a plus, so that you can more reliably detect and attack virus. A lot of the data that I had just showed you was with a viral surrogate that's a bacteriophage that's chosen because it has the exact same physicochemical properties of human noroviruses, so same capsid organization, size, charge. You would expect it to behave similar to norovirus, but we also wanted to just confirm this with other more genetically related eukaryotic virus surrogates. So on the left there, you have murine norovirus that you again see, mostly pretty positive results. And then the same with Tulane virus, which is in the same, it's in the same family as human noroviruses, whereas murine norovirus is in the same genus. They both display sort of similar capture efficiencies and recoveries. And one of the other things that we were really interested in, because again, as I mentioned sort of in the beginning, is that we saw and were able to observe these mills being able to capture free RNA, was seeing, are these mills compatible with RNA extraction buffers? And we do see similar or comparable performance compared to like the traditional bead-based, silica bead-based extraction, things that you might use in a viral extraction kit, which is exciting because that potentially enables these mills to be used as a single tube capture and extraction reagent. So it's sort of a one-stop shop. You take your food that may contain the virus, you can capture the viral particles, rinse away residual food, add your extraction buffer to release the RNA from the virus, and those mills will recapture that RNA. You can wash again and then elute the RNA off. So you pretty much take your food input into the tube, and then you end up with the RNA ready to go for PCR. So this is really exciting work that Sloan Stouffer did, and we're continuing to improve upon it. In addition to magnetic ionic liquids, we're also investigating the use of another type of magnetic liquid called deep eutectic solvents. These are a little bit newer and they're really exciting in sort of the magnetic liquid field because they are generally considered greener and potentially cheaper to synthesize, and you can synthesize it more readily to scale. These deep eutectic solvents consist of a hydrogen donor and acceptor, and they also display paramagnetic properties similar to the magnetic ionic liquids. Again, there's their chemical structures. And as you'll note as well, they also, again, have like a metallic core associated with them, or an anion associated with them. And we see with these DESs similar, comparable recovery with the DESs compared to the mills, which is what we expect. And this is really awesome work done by Lily Sayad. And similar to the mills, we see comparable recovery efficiencies with DESs, again, confirming that DESs don't necessarily display antiviral properties. This was really interesting for DESs because some of the components of them have actually been in isolation shown to be antibacterial. But it seems like when they're actually formed into the DES itself, they at least for viruses, don't display any antimicrobial effect. So that sort of talks about sort of the upstream. Again, it's a major roadblock, upstream sample preparation that you need to get those small number of viruses out of your food sample. We also have a number of pretty exciting the downstream detection methods. So we've concentrated the viruses. How do you potentially detect it? Generally, RT-qPCR, which I'm sure everybody's heard about now with the pandemic, tends to be the gold standard for detection of viruses in foods, in particular, again, because viruses are at low levels and RT-qPCR has really good analytical sensitivity. So that's why it tends to be used compared to some other ligand-based methods. Generally, ligand-based methods, I'm sure we're also familiar with the pandemic. If you've ever used those sort of at-home testing kits where you swab your nostril and then put it in the water and use a lateral flow stick, those are ligand-based. They generally tend not to have as good analytical sensitivity. So that's why RT-qPCR is the gold standard for detection of viruses in foods. But there are potentially some limitations to RT-qPCR. There are portable instruments now that you can buy, but it is prone to inhibitors. And it also is going to be limited in how much you can subtype, which I'll talk about in a minute. So we have a really awesome researcher here at UMass, Dr. Min Chen, and the Department of Chemistry, who does a lot of really, really cool work with nanopore sensing. So some of you may have heard of nanopore technology in the context of nanopore sequencing, which is also really cool. This is not necessarily that. This is a different type of nanopore called quiet outer membrane protein G. And in the past, Min has showed that this is portable, it's reliable, you can really quantitate what you're trying to detect, and there is some tolerance of inhibitors. but there's also some other properties of it that maybe suggest that you can get a very strong fingerprint signal and therefore subtype, which is really important, especially for pathogens that are very diverse like noroviruses and very prevalent. In order to identify a cluster of cases in an outbreak or potentially attribute a direction of transmission, you generally need to subtype below just the genus level of saying, oh, it's norovirus, or this person has norovirus, this person has norovirus. You really want to have a strain level designation to link them together, if that makes sense. And there are inherent limitations in the way we can do that for viruses, like with whole genome sequencing in particular, the amount of time it takes, as well as the amount of input genetic material that you need for it. Whole genome sequencing isn't necessarily ideal for viruses and foods. And so the way that this nanopore sensing works is similar to a lot of other electrochemical sensing techniques, where you're essentially measuring ionic flow across a membrane. So you have this pore protein, which embeds itself in the membrane. It's a porin that's on the surface of E. coli. And you measure the ionic flow across the membrane. And when something interacts with it, it'll create a sort of fingerprint signal in a number of ways, sort of visualized there in terms of the intensity of how much it's disrupting the flow of ions, the dwell time, what they call the dwell time, how long it's staying in the pore and disrupting it. And in addition, there's something that I'll show you in the next slide that in particular this OMG protein does is it exhibits something called gating behavior. So you get another even level of resolution in terms of there's a back and forth flow that creates a little bit of noise. And when that noise gets disrupted, it creates another sort of metric that you can measure to create sort of a fingerprint signal. So again, if we had a more portable technique that was able to not only detect that it's norovirus, but also subtype, it could be really, really important for identifying outbreaks and potentially attributing a source of transmission. And if it can do it not only portably and rapidly, but also sensitively, where you don't require as much input virus, again, because viruses, when they contaminate foods or the environment, can be at low levels, then that would be really kind of a game changer. And so that's why we're really excited about this technology. And so right now, this is really awesome work done by Minji Kim and also Shang Yu. I'll show you some of the work he's doing too right now currently. We did a little bit of work, in this case, using a flag tag. I'm not sure if folks have heard of that, but it's a surrogate target that we used, which we know we have a flag tag and we know we have anti-flag antibodies that'll actually bind. So we're using this sort of as a model to understand, the influence of where you put the bait flag sequence on that loop that I just mentioned, and how that influences your ability to detect the antibody or the target, as well as the potential for this technology to subtype if you can't get it to work. And luckily we were able to find you can actually produce optimal levels of noise when you locate here, sort of visualized in this set of images. And then once we were able to optimize sort of the background noise, again, the noisier it is, the better, because you can actually create more of a fingerprint when it disrupts the ionic flow, if that makes sense. And we found out, luckily, that not only could we detect different proteins, you could subtype them at a very, very specific low level, or very specific level. So here are 3 different monoclonal antibodies, and we could get and produce a unique fingerprint signature that we could pull out repeatedly from all 3. Which again suggests that there could be really, really powerful subtyping ability, similar to like what you would see with mass spec. Not only that, you can actually pull out these fingerprints when you use a mixture of these monoclonal antibodies, as sort of visualized here. You have FGF4R and OTI4C5 that you're able to pull out in a mixture. And then on top of that, it's so sensitive and specific at fingerprinting, you can actually pull out different antibodies in a polyclonal mixture of antibodies as well, which is really exciting, again, suggesting that it has very, very powerful fingerprinting subtyping ability. And then, as I sort of alluded to earlier, one of the questions you always want to ask when you're trying to talk about detection in foods is how well can this technique withstand sort of more complex matrices. So we overloaded the system with a lot of fetal bovine serum, which is a lot of competing protein compared to the antibody, and we're still able to pick out signatures suggesting that there is an ability to tolerate matrix-related inhibitors. which is also really promising. So our next steps with this, in addition to trying to optimize electroosmotic flow, is to try and develop it for viruses. So it shows really quite a bit of promise to be used as a detection subtype technology for viruses. One thing that I just mentioned is really cool work that Shang Yu and Joshua Foster in the Min Chen lab are working on, is trying to enhance the sensitivity further of the technology by increasing something called the electroosmotic flow. Essentially, the flow of the ions through the flow of the sort of current through. The idea is if you can increase that flow, it's going to pull whatever your target is to the pore and through the and into the pore more quickly. So you're going to have quicker and more higher sensitivity because you're going to sample much more of the liquid in a quicker time. And the idea that they're doing that is they're trying to change the charge in the bottom of the pore. I have a pretty good image in the next slide here, where you're trying to sort of make amino acid mutations without completely destroying the structure of the pore in itself, to try and pull those ions through and create more of a stronger electroosmotic flow through the nanopore to potentially enhance sensitivity as well as time detection. And Shang's done a lot of really cool work here, creating a whole bunch of different mutants, where he's trying to, again, alter the amino acid profile, in particular the bottom portion of that porin, to try and pull the buffer through, and has been able to achieve a pretty good improvement from 1.18 to 1.44. And so he's continuing doing this really cool work. And we're also in the process of starting to try and think about really applying this to more viruses. Now we've done it to the server. So the last technology that I'll talk about related to detection is something called molecularly imprinted polymer nanoparticles, or nanomips. This is an exciting technology because these nanomips are extremely stable in quite a variety of conditions. So a wide range of pHs, these polymers can sort of maintain their stability as well as high temperatures. So there's some past reports. This is in collaboration with a lab that was at Newcastle University, is now in University of Manchester in the UK, where they demonstrated that these could maintain sort of affinity for their target even after autoclaving. So extreme thermal intensity as well as ranges of pH can be withstood by these nanomips, which is really nice because not only does it make it low cost, it means you can potentially regenerate the device that you're using by adding like a harsh buffer to elute off whatever your detection target was, so that you can reuse it, so it lowers cost. So we decided to see how well this sort of technique would work for noroviruses. So essentially the way that molecularly imprinted polymers work is kind of like you make a jello mold against your target. So in this case, we took a norovirus peptide, a 10-mer amino acid sequence, put it on glass beads, and then you add a monomer and you sort of polymerize that monomer around your target, and it's left with sort of an indent of your target that then can specifically bind your target. And so these haven't really been created for especially non-enveloped viruses. One of the other things that's kind of exciting that I won't get into too much is that we were able to just order a 10-mer synthesized peptide and use that as a target, as opposed to having to create virus-like particles, which is the whole assembled capsid, as a target, which is traditionally what's used to create affinity ligands, and can be very cumbersome to create, purify, versus if you can just get a chemically synthesized 10-mer peptide and use it, that would be really big. So we chose a 10-mer peptide. We picked from the P1 subdomain of the viral capsid. So up in the upper left corner there, you have a dimer of this capsid protein that forms the viral capsid. And that domain is broken up into the P2 and P1. The P stands for protruding. It's sort of those spikes on the surface of the virus. P2 is what interacts with the host receptor as well as a lot of the antibodies. And it's not very conserved. It's super hypervariable because it's the thing that's mutating the most when it's trying to escape host immunity. And then as you get down further to the core, or the sort of surface of the viral particle, you end up with the P1, which is a little bit more conserved. And so we picked a little bit more conserved of a target, with the idea that, because these viruses are so diverse, traditionally with ligands that we use for sensing, the challenge is coming up with ligands that are broadly reactive against all of the noroviruses. Like they may only bind one set of strains of the virus, and not be able to bind others because they're so genetically diverse and structurally diverse. So that's why we picked the P1. And we were able to create pretty well-performing nanomips. This is in buffer, where not only were we able to detect the peptide, but we were also able to detect full assembled viral particles. Again, which is a really promising result, showing that you can just get away with using a 10-mer amino acid peptide as your target for creating ligands versus having to use a full assembled viral particle. We're able to get cross-reactivity as well, which is, as I mentioned, really, really exciting. So it's pan-genogroup. So genogroup is the broadest sort of way that we subtype noroviruses. And we were able to get cross-reactivity against very, very diverse strains of VLPs relative to the genogroup that the nanomips were created against, which again suggests extremely broad reactivity. So you see we get good reactivity against G11 and G24 viral particles. We did some further, this is really awesome work by Amy Dan at the University of Manchester, where she looked even further in terms of genetic relatedness. And it's good and bad that we see sort of not only reactivity across the two different genogroups of human norovirus, but we also see, although reduced, we do see a certain level of reactivity with murine norovirus, which is genogroup 5, and then Tulane virus, which is a different genus, but in the same family. Luckily, with bacteriophage MS2, which is very, very genetically distant, even though it's structurally similar, it's very, very genetically unrelated. We don't see really very much reactivity with that. And so this suggests that you kind of have the inverse problem with these molecularly imprinted polymers for viruses, is that you actually end up with really broad reactivity. So going forward with this, we want to try and tune the target peptide that we're using to create these closer to the P2 subdomain so that it's a little bit more specific to human noroviruses. Though there is also application of if you detect any type of coliciviridae, it's a virus, generally not good for anything. So, or for anyone. So there's also value there. But that is really exciting work, again, because you kind of have the opposite problem with these MIPS where you get really, really broad reactivity, which is generally really good for most applications for noroviruses. So finally, the last thing that I'd like to sort of talk about is sort of the control element of some of the really cool work that we're doing. And so there's some work here that was done by Anand Srinidhi. He's now at University of Missouri. And this was in collaboration with Dr. Philip Democritou, who's now at Rutgers. And this involves an electrospray ionization-based technology that they call engineered water nanostructures. It's considered a dry technology, which is really cool. So essentially the way that it works is you take sort of a number of potentially antiviral and antimicrobial ingredients, and then you basically run them through a charged needle that creates very, very small nano-sized water particles that contain these different ingredients. And it's considered dry because those particles are so small, they almost immediately evaporate upon landing at your target. This process also creates a lot of sort of reactive oxygen species, in addition to potentially more effectively delivering these specific compounds that you use to your target. And so it can enhance the activity of it and simultaneously use a lot less of this sort of active ingredient than you would otherwise, like with a spray bottle. The advantage of that is not only are these potentially more green or greener inactivation agents, but that you leave less residue after you apply it. And that is an area of concern, especially in environmental health, the amount of residual residue that's left from some of the disinfectants. I don't know if this will play, but there's actually, as you can see, you actually have to keep a camera trained on this. I don't know if you can see my mouse, but this is the needle. You take your solution, you run it through it, and it creates this invisible sort of stream. So you have to use a higher power camera to sort of observe if it's on or not. And again, because it's dry, you kind of put your hand under it, it'll still be dry as you apply it. So we did this. Again, it had been really demonstrated a lot for bacteria as well as enveloped viruses like influenza virus. Hadn't been investigated against these foodborne viruses, which tend to be non-enveloped and tend to be a little bit harder to inactivate. And so we were able to show both on surfaces like stainless steel, as well as on a more complex matrix. Here it's on a spinach leaf that I'm sort of displaying the graph there. We see pretty promising inactivation for a number of these cocktails of the sort of active ingredients. And the even more exciting thing is that you're really delivering quite a low level of those active ingredients onto your target, and you're still getting pretty good reduction of norovirus and of a norovirus surrogate, bacteriophage MS2, which you mentioned earlier. So it's a pretty cool technology that we're still continuing to explore. As you can imagine, there are many different combinations of cocktails and ways you can formulate them. And so it's something that we're interested in continuing. And finally, lastly, I'm just going to introduce the concept because we don't have any data for it at this moment. But Julia Fukuba, who you mentioned earlier, is doing some really cool work related to understanding the potential of noroviruses in particular, or norovirus surrogates to develop enhanced recalcitrance. So a lot of people have heard of antimicrobial resistance with bacteria. This is totally a different mechanism because viruses aren't living. But one of the things that we noticed is that, a lot of these inactivation agents people are using tend not to display the most effective inactivation of virus, on top of the fact that you have a lot of human behavior when people apply disinfectants that's not really putting them in their best foot forward. So you don't, a lot of times people don't maintain proper contact time, and a lot of times people don't clean before they disinfect. So there's a bunch of what they call organic load, that'll also react with your disinfectant and reduce its efficacy. So we believe there's potential for a lot of subfatal treatments of these viruses to be applied in the environment. And given how prevalent these viruses are and how rapidly they evolve with high error rates, that there could be some selection pressure for creating variants of these viruses that are more resistant to disinfectants. And so there's a really cool lab in Japan that did 2 papers with this, looking at one viral surrogate with 2 different disinfectants, and demonstrated that you can, in certain instances, get variants that are dramatically more resistant to inactivation after serial subfatal treatments. So the picture on the right is from one of those papers with chlorine. And so we were sort of wondering, well, does the same phenomenon occur with some of the other disinfectants that are commonly used, like alcohol, quaternary ammonium, and peroxyacetic acid, that you can see used in some cases in food settings. And then on top of that, we have some other hypotheses that I'm not going to talk about, where we're trying to more fundamentally understand the influence of dynamics of the viral capsid protein on resistance to inactivation. If we do happen to observe variants that are more resistant, we can then conduct structural analysis on the dynamics of those capsids to see how much the molecular dynamics of the capsid potentially influence their resistance to inactivation compared to the wild type. And Julia Fukuba, shout it out, is doing really, really cool work related to that. And so, sort of to conclude, we have some really exciting projects related to concentrating these viruses, so solving that needle in a haystack question using either engineered bacteria or magnetic liquids. We have some really cool work looking at detecting and subtyping these viruses in a portable manner using the OMCHI nanopore protein, as well as nanomips. And then we have some interesting projects related to better targeting these viruses, or better formulating disinfectants to target these viruses, as well as understanding the fundamental structural dynamics of these viruses that make them more resistant to inactivation, and their potential to form or to develop more enhanced recalcitrance to those disinfectants. So I wanted to thank everybody here for taking the time and listening to me drone on about all that for 45 minutes. And I especially wanted to thank all of our awesome lab members. I know a number of you folks are here, as well as our past lab members who've moved on, graduated, who generated all the data, and I get to give a talk here and take credit for it, as well as all of our awesome collaborators. And in particular, also would like to thank our funding support from the USDA, NIFA, NSF, and the Massachusetts Department of Agricultural Resources. And that's pretty much it. So I'll take any questions if anybody has any. And thanks again for attending and listening to some of the cool work that we're doing.
Thank you, Dr. Moa, for the wonderful presentation. We have time for a couple of questions. Any questions from audience?
It could also just be generally about diarrhea, if anybody has any general diarrhea related questions. That is one thing I didn't mention. We always are looking for diarrhea. So as I sort of mentioned, we don't have a really good culture system. So the only way we can get infectious virus is to get our hands on some stool, human stool that contains the virus. So we're always looking for diarrhea. Obviously not like you'd have gloved hands. That's a good question, Rachel. Actually, when I was in grad school, it was a shame because I was in North Carolina. The folks at Emory, we actually work with them, Juan Leon, actually do the feeding study. So one thing that people don't know about norovirus is that you can actually do human feeding studies with it. And at least, but this was like a while ago. It was probably almost a decade ago. I think the going rate was like 1,500 that you could do it if you qualified. So if anybody wants to make $1,500, maybe reach out. Or if you just have diarrhea, let me know so that we can try and get some.
Okay, talking more, I know you are very successful in doing research, and you receive uh, prestigious awards, including IFT Outstanding Young Scholar Reward, and also Phi Tau Sigma Outstanding Young Achievement Awards. Can you share? I know many of the audience are students. Can you share with us your experiences in terms of how to do productive research?
Yeah, thanks, Fanbin. That's a good question. I think it all starts with being able to find amazing students. I have to say, like, I think there is some element of luck, and I've just been lucky to have really, really awesome students that I get to work with. I'm not just saying that, to kiss up because a number of them are here, but I really mean it. Like, they actually are really, really awesome students. And so I think that's really key to it. So like I just kind of get to take credit for it and get the awards. But really, when you have really awesome students like we've been able to have, that really makes like a night and day difference. Um, I would say I think just yeah, I would just say like especially if you're interested in going into research, like as like a professor, like a research teaching professor, really making sure that what you do research on is something that you're genuinely really interested in. Because as we were talking about earlier, Fan Bin, like having to go through the slog that we're going through now of like writing all these grants and getting all that, like if your heart's not in it, it's really it can be a little bit deflating. So I would say really making sure that you can find really awesome people. There's generally going to be really awesome science that follows. And also really being passionate about what you're doing research on, not just doing it for the sake of getting publications or that type of stuff. So really like the awards are really for the all the work that my students have done, not really me. That's a Do I work with undergraduates is a great question. We do, a number of the folks I listed in the acknowledgement slide are undergraduates. We have a number of really awesome undergraduates right now. So Shafina Uden, who's doing some work helping out with the mills and the DESs that I mentioned, as well as Ashlyn Binu, who's doing some really cool work with bacterial concentration. A number of my lab members here. That's one of the other things that's really helpful is like, again, when you have really awesome graduate students, they do a really, really good job of mentoring the undergraduates, and it's really rewarding to see them accomplish a lot of really cool stuff. So we do definitely work with undergraduates. That's one thing that I think is extremely valuable. If there are any undergraduates here, um, getting to do some research and really figuring out like how much you like research, especially if you're thinking about grad school, that'll give you an idea of like, hey, do you want to go straight to grad school or do you maybe want to go to industry first? Doing undergraduate research is extremely valuable in addition to like, industry internships.
That's cool. I also noticed that you have collaborations with many the people in different area, chemistry and people in it's not a traditional food science. So that's what I
Yeah.
say. How did you develop this kind of collaborations?
That's a really good question. That's one thing that I really learned from my PhD because I was working with Leanne Jacobs at NC State, and she was like sort of head of this huge $25 million USDA collaborative grant. And so she set a really good example of like how to reach out and work with people. I think a lot of it is luck. And that's one of the other cool things I think in general about food science, and especially like I can only really speak to food safety. But it's a very collaborative sort of subdiscipline or discipline in general, at least from what I gather from other, my colleagues, which is really like we are really lucky to be in food science because it is a very collaborative sort of field. So people are always really willing to work with you and reach out. I would say like, yeah, like I like actually meeting Min Chen was just luck. Like I was talking to Julian in our department. We were like having a beer one day, and I was talking because I was interested at the time in the nanopore sequencing, and he said, "Oh yeah, I think there's somebody who does nanopore here at UMass," and that's real. And I just emailed her, and that's how we've had like one of these extremely fruitful collaborations and this really exciting technology that developed out of it. So some of it's just really luck. Um, Same with like getting to work with like some like Phil or Marlos, who's the folks who do the nanomibs. A lot of it is just somewhat luck. Yeah, so yeah, undergraduates undergraduates really do a good job with research. And it's always rewarding to I think it's important personally to have my graduate students mentor at least one undergraduate, because I think that's also a really good skill set, especially if they're getting like a PhD or master's. They're going to have people working under them when they go into industry or government or academia. So getting that experience is really good. So it's a two-way street. So the undergraduates do a really good job in research, but it also really helps our graduate students out as well.